Thank you, Christian. So in the work that I will present today, we applied connectopic mapping to investigate dopamine-specific projections in the human striatum. And when considering the dopaminergic system in the human brain, there are basically two main dopaminergic pathways that both connect to the striatum. The first one is the nigrostriatal pathway, indicated in red in the image. And this pathway consists of dopaminergic neurons projecting from the substantia nigra to the dorsolateral striatum, including the dorsal putamen and caudate. And this pathway has mainly been associated with motor function. The second pathway is the mesolimbic pathway, indicated in green, and this consists of neurons projecting from the ventral tegmental area in the midbrain to the ventromedial striatum, including the nucleus accumbens. And this pathway has predominantly been related to reward-seeking behaviors. Alterations in these pathways have been implicated in several neurological and psychiatric conditions, including substance abuse and Parkinson's disease. And in this talk, I will mainly focus on Parkinson's disease, which is a neurodegenerative disorder characterized by impaired control of body movements, such as tremor and slowness of movement. The disorder is caused by loss of dopaminergic projections in the substantia nigra. And because these neurons project to the striatum, this means that there's also a loss of dopamine in the striatum. This occurs first in the putamen, but later also in the caudate regions. The disorder is usually diagnosed using a clinical assessment, but sometimes also a DAT SPECT scan is used to aid in diagnosis. And when patients undergo such a DAT SPECT scan, they are first injected with a radioactive tracer, which binds to the dopamine transporter in the brain. The dopamine transporter is present on the presynaptic neuron, where it removes dopamine from the synaptic cleft after it has been released. And therefore, the dopamine transporter can be seen as a marker of dopaminergic neurons. Well, on the image on the bottom of the screen, on the left side, you see a DAT SPECT scan of a healthy participant. On the right side, a DAT SPECT scan of a patient with Parkinson's disease. And you can clearly see the reduction in the DAT signal coming from the striatum, which is thought to reflect the reduction in dopaminergic neurons. However, the investigation of the dopaminergic system in vivo in the human brain is quite problematic. Although a DAT SPECT scan can be used, it's of low resolution, it's invasive, and it's also not possible to investigate acute changes in dopaminergic functioning. So most of our knowledge is actually still based on animal work, because also with current fMRI methods, it's not possible to isolate these dopaminergic projections. And this is especially relevant for the striatum, which is highly interconnected. It does not only receive dopaminergic projections, but also other types of projections. However, in this work, we aim to use connectopic mapping to investigate these dopaminergic projections in the striatum in resting state fMRI data. To this end, we first investigate whether there is a relationship between the gradients obtained with connectopic mapping and the DAT SPECT scan. And later we investigate whether these striatal gradients are also altered in Parkinson's disease, the disorder characterized by alterations in dopaminergic functioning. So we made use of connectopic mapping, the method that Koen has just clearly explained, so I won't spend much time on this slide. But importantly, our region of interest is the striatum. And in the end, for the Parkinson's disease study, we will also apply transsurface modeling to summarize the gradients in a small number of coefficients with which we can do the statistics. So as Koen has already explained, the first gradient in the striatum represents the anatomical subdivision into putamen, caudate, and accumbens. The second gradient is a ventromedial to dorsolateral gradient associated with corticostriatal goal-directed behaviors. But there is also this third gradient. And as you can see, this gradient is very different from the first and second gradient. So we wanted to investigate whether this gradient could maybe be related to dopaminergic projections. To this end, we selected DAT SPECT scans from 209 controls of the Parkinson Progression Marker Initiative, which is a very large multi-center data set that's publicly available, and that includes several types of data, including DAT SPECT scans from Parkinson's disease patients and controls. And here we selected the DAT-SPECT scans of all the controls, averaged them, and masked them for the striatum. The resulting average DAT-SPECT scan is shown here. And the red voxels in this image indicate a high amount of DAT binding, which is thought to map on a high density of dopaminergic projections. When we then compare the DAT-SPECT scan with the third striatal gradient obtained by applying connectopic mapping to resting state fMRI data, we can see the huge correspondence between the two. The actual overall spatial correlation is 0.88, so very high. So what we've shown here is that the third striatal gradient obtained from resting state fMRI data maps onto the DAT-SPECT scan and therefore maps onto dopaminergic projections to the striatum. A thing to keep in mind here, though, is that the DAT-SPECT scan and the gradient were obtained in different data sets. For connectopic mapping, we need a relatively high resolution resting state scan, especially since we're now looking at the third mode of connectivity. But to our knowledge, there's no data set available that includes both a high resolution resting state scan and a DAT-SPECT scan from the same participants. So our results do await within subject replication. But nevertheless, this correspondence looks quite convincing. So then as a next step, we investigated whether this gradient was also altered in Parkinson's disease, since Parkinson's disease is associated with a reduction in dopaminergic projections. Although the PPMI dataset has some resting state fMRI data available for some of its participants, it's of a relatively low resolution. So we decided to make use of a different dataset here, including high resolution resting state fMRI data for 20 Parkinson's disease patients. All these patients had 2 sessions. During one session they received levodopa, during the other session placebo. And levodopa is a precursor of dopamine that is often used for the treatment of Parkinson's disease. So we had a resting state scan available for both sessions, and also during both sessions the Unified Parkinson's Disease Rating Scale was administered, which is a measure of symptom severity. So we applied connectopic mapping to the resting state data of both sessions. We applied it separately to the putamen and the caudate nucleus accumbens region, because Parkinson's disease is known to affect the putamen region first, before the caudate, and hereby we increased our regional specificity. First, we investigated whether a relationship with symptom severity was present. And then we also looked at the effects of acute dopaminergic modulation by investigating whether there was a difference in the gradient between the placebo session and the levodopa session. And we also conducted a more fine-grained analysis, investigating whether the reduction in symptoms caused by levodopa compared to the placebo session was also accompanied by a change in the TSM coefficients modeling the gradient. We conducted so-called omnibus tests, testing all the transsurface coefficients modeling the gradient at once. And if we then found a significant difference or a significant association, we tested the individual TSM coefficients. And in this way, we can maybe explain in which direction the gradient changes most. And we did indeed find association between the third striatal connectivity mode and symptom severity in the putamen, but not in the caudate region. Postdoc Pearson correlations revealed that especially the quadratic coefficients modeling the gradient in Y, so anterior-posterior direction, and Z, superior-inferior direction, contributed most strongly. This figure shows the relationship between symptom severity and the Y parameter. We then selected the data points closest to the regression line at different symptom severity scores. And when we then plot the corresponding gradients to these data points, we can see this very nice association, which shows that at higher symptom severity, the amount of blue voxels increases, the amount of red voxels decreases, And because we have just mapped this gradient onto the DOT SPECT scan, we can infer that this represents a reduction in the amount of dopaminergic projections, which is pretty nice and which is also what we would expect. We also investigated the effects of acute dopaminergic modulation. However, we didn't find a significant difference between the placebo session and levodopa session. But we also know that not all patients with Parkinson's disease respond equally well to levodopa treatment. So therefore, we conducted a more fine-grained analysis, investigating whether the change in symptom severity induced by levodopa compared to the placebo session was accompanied by a change in the TSM coefficients modeling the gradient. And we indeed found such an association, again only for the putamen region, not for the caudate. This association is shown here, and what it basically indicates is that the larger the reduction in symptoms caused by levodopa, the larger the change in the transsurface coefficients modeling the gradient, so the larger the change in the gradient. So what we've shown here is not only that this gradient is different in patients with Parkinson's disease, but also that it's sensitive to acute dopaminergic modulation. So to conclude, we have shown that the third striatal gradient obtained with connectopic mapping maps onto the DOT SPECT scan. Hereby we have found a non-invasive method for investigating the dopaminergic system. We have also demonstrated that it can be used as a biomarker for altered dopaminergic signaling. We have shown an association with symptom severity in Parkinson's disease. We also showed that it was sensitive to acute dopaminergic modulation. And I didn't show the data here, but we have also found an association with the amount of alcohol use and smoking in the Human Connectome project dataset. So alcohol use and smoking is also associated with alterations in the dopaminergic system, so that's what we would expect. So, and importantly, it's really the fact that connectopic mapping allows for the detection of multiple overlapping connectivity modes, so it allows for functional multiplicity, that we were able to isolate these dopaminergic projections. To conclude, I would like to thank the team from the Donders Institute, but also my current lab, led by Alex Fornito at Monash University. And I would like to thank you for your attention.